So we were talking about the secondary structure of proteins, and this is the beginning of Chapter 5, Part 6 of our lecture. And so the presence of either the alpha helix or the beta sheet is going to occur in specific regions of a polypeptide chain, and the type or the function of that protein is actually going to be facilitated by how many alpha helixes or beta sheets there might be. So an example of this I was starting to talk about is that keratin, which is found in the hair, has a lot of alpha helixes, and these alpha helixes occur throughout the most of the length of that protein. And in this case, it helps it to get that long, fibrous shape that you can see with hair. So the next level of structure is a tertiary structure. And this is determined by the interactions among the various R groups. So again, the secondary structure had to do with the interactions of the amino and the hydrogen in the amino groups and the oxygens in the carboxyl groups having hydrogen bonds. Now, the tertiary structure does include hydrogen bonds as well, but there are also bonds between polar or charged areas of the R groups. There are ionic bonds, and there are hydrophobic interactions, and there are also van der Waal interactions among R groups. So all these different types of bonds work to make the tertiary structure. The last type of bond is a really important one, and that's the disulfide bridges. And those form again between the sulfhydryl groups, the R groups that have sulfhydryl groups of two cysteine monomers can act to rivet parts of the protein together. So the disulfide bridges are very strong covalent bonds, and they are critical to the ability of a protein to hold together because they are the strongest. All the other types of bonds we've been describing are very weak, but cumulatively they do form the structure, the important structure of a protein. But these disulfide bridges become very critical since they are so very strong. The last level of structure is the quaternary structure. And so we didn't mention this before because this involves more than one polypeptide chain. So up until now, the primary, secondary, tertiary structures are all one polypeptide chain in its interactions with itself. But the quaternary structure is going to be the interaction with another polypeptide chain. And so this results from the grouping together of two or more polypeptide subunits, for example, like collagen, which is the fibrous protein that makes up our skin, or is a big part of our skin. And it's made up of three polypeptides that are supercoiled into a larger triple helix. And that helix provides a structural strength for collagen in terms of its connective tissue character. And it actually makes it as strong like a rope, enabling our skin, for example, to be strong or flexible. You also find collagen in your tendons or ligaments. It helps those to be strong and flexible. Another example of this quaternary structure is in a hemoglobin molecule, which is made up of four polypeptide subunits. It ends up with two alpha and two beta chains, and both of these consist primarily of alpha helical secondary structures. Each of these subunits has a non-peptide heme component with an iron atom that binds oxygen, and so that allows hemoglobin to carry out its job of carrying oxygen around in the blood. So in review, the sequence of an amino acid chain determines its function, or it actually determines its structure, which then determines its function. If you change the sequence of amino acids, you're going to change the structure, and therefore you'll change the function of your protein. So any polypeptide chain will have a given amino acid sequence that spontaneously arranges itself into a three-dimensional shape. And that shape is going to be determined and maintained by the interactions responsible for either the secondary or tertiary structure. And this folding occurs within the cell. So just as we can have it fold within the cell, we can also have it unfold. So if we alter pH, salt concentration, which is an ion, temperature, and there are other factors, these things can cause the protein to unravel or what is called denature. And what happens is that those forces disrupt the weak chemical bonds and interactions within the protein that are holding it together. And so when it becomes misshapen, the denatured protein is no longer active, it's biologically inactive. So most proteins are going to become denatured if they're transferred from the aqueous, the water environment of a cell, to a nonpolar solvent. So for example, one of our lab groups used oil, which would be act as a nonpolar solvent. What happens is the polypeptide chain refolds itself so that its hydrophobic regions now face outward instead of normally they would be facing away from water. Now that they're in a hydrophobic environment, they're actually going to, or a nonpolar environment, they're going to actually reshape and flip inside out. Other denaturation agents are chemicals that disrupt the hydrogen bonds, which will affect the secondary structure, or ionic. There are things that disrupt the ionic bonds or disulfide bridges, and that's going to disrupt the tertiary structure. When we have heat, denaturation occurs because it disrupts the weak interactions that stabilize the structure. And that is a reason why high fevers can be fatal, because what happens when you get your high fever is we have denaturation of proteins, particularly enzymes, so that cellular processes come to a stop. After denaturation, most proteins can return to their original shape. And this fact suggests that the information for building a specific shape is intrinsic in the protein's primary structure. Right now, biochemists know the amino acid sequence of about 24 million proteins, and you can check this by going to the protein database on the internet and seeing what the three-dimensional shape looks like. The three-dimensional shape is actually only known in about 25,000 of those 24 million proteins that we know the primary structure. But it's difficult still to predict the three-dimensional shape of a protein. And there's actually a place online where you can go and help try and predict the shape and work out the shape of a protein based on its primary structure. So again, we know 24 million proteins primary structure, but we only know 25,000 of the tertiary or quaternary structures. So there is a site online where you can go and try and come up with the structures yourself and try and suggest what the three-dimensional shape should look like. The folding of the proteins is assisted by a protein called a chaperonin, or the chaperone protein. And these don't specify the final structure of the polypeptide, but they work to segregate and protect the polypeptide while it's folding spontaneously. So this is what they look like, actually. This is a chaperonin. It has a cap region, and it's like a tube. And so the polypeptide chain will go into the chaperonin and do its folding within. The cap goes back on, which causes the chaperonin actually to change its shape. And then the polypeptide, in some way, is being assisted within this structure to get into its final three-dimensional shape, or its tertiary structure. Another thing is that cells interact with chaperonins to mark incorrectly folded proteins, so that they get refolded or they get destroyed by the cell. So that's a pretty mysterious and interesting function of these proteins, chaperonin proteins. If you get too many incorrectly folded polypeptides, you actually end up with a number of diseases. That one of them is Alzheimer's, another is Parkinson's, mad cow disease is another. Each of these have in common a large number of incorrectly folded polypeptides. This picture here shows you RNA polymerase. RNA polymerase was in 1969 just thought of this glob that goes along DNA, making the messenger RNA molecules. Then in 1991, this is the structure that was determined for RNA polymerase. And then just in 2001, this is the actual sequencing, both the primary and secondary and tertiary structure of RNA polymerase. So now on to lipids, fats, and oils. So this is our last biomolecule. And we left this one for last because this is the only one that really isn't actually a polymer, because it's not a series of monomers joined together, but actually is formed from a number of subunits that are different. The function of a lipid in general is long-term energy storage and very concentrated energy, and we'll see how that occurs with its structure. So first of all, it's formed through dehydration synthesis, just like other biomolecules that are actually polymers. But in this case, it's a ester linkage between a glycerol molecule and a fatty acid chain. So it's actually two different types of molecules that are being joined together through dehydration synthesis. So this ester linkage is between a hydroxyl and a carboxyl group, again forming our water molecule. And you can, with one glycerol molecule, you can have either one fatty acid chain attached, two, or up to three. So glycerol is a 3-carbon alcohol with a hydroxyl group attached to each of those 3 carbons, which allows it to make those 3 bonds to fatty acid chains. The fatty acid is consisting of a carboxyl group, which is again allowing it to make this bond, dehydration bond. But after that, it's a series of hydrocarbons, so a long carbon chain with hydrogens attached. These many nonpolar carbon-hydrogen bonds are what make a fat hydrophobic. So as you remember, this type of carbon-hydrogen formation is not reactive. It's a very stable molecule. It is very hydrophobic. So this is what allows this molecule to be such a great storage molecule and to really be densely storing energy because of all these carbon-hydrogen bonds here. when we have three fatty